Chapter 4 Biological Molecules. Again, the objectives are very important. By the end of the lesson, go back to the objectives and make sure that everything that you're supposed to do, you can do. Okay, most of the food we eat is grouped into three main major categories. Those categories are carbohydrates, proteins, and fats. And actually, those are the three main categories that our body needs also. So, everything that we eat will always go into a function that our body needs. Let's take a look into carbohydrates. So, carbohydrates, they are made of three main elements: carbon, hydrogen, oxygen, in the ratio of 1 to 2 to 1, which means that the amount of hydrogen is double the amount of carbon and also double the amount of oxygen. So as you see here in the glucose molecule, it contains 6 carbon atoms, therefore it has 12 hydrogen atoms, and oxygen 6. So this is simply the composition of carbohydrates. Let's take a look about its shape. So the shape of any carbohydrate molecule, it looks like a crystal, or it has a hexagonal shape, like the shape that you can see here. The simplest type of carbohydrates are monosaccharides. Monosaccharides are sweet and soluble in water. Like what? Like glucose, fructose, the sugar of fruits, and galactose. Simple sugar molecules join together to form a disaccharide. And as we established before, di in biology means two in Latin. Okay, so sucrose, maltose, and lactose, these are disaccharides. When you put two sugars or two mono, and again, mono means one. So when you put two monos together, you get a di. So monosaccharide and the monosaccharide, you get a disaccharide. So when you put a chain of glucose molecules together or a chain of monosaccharides together, what do you get? You get a carbohydrate, which is a polymer. So we call this carbohydrate a polymer. Okay? While the monos, these are the monomers. So like what? Like starch, glycogen, cellulose that makes the plant cell wall, remember? So the cell wall, it's made of cellulose, as we said before. Okay, so you already know one of the importance. It makes the cellulose, which builds cell walls, which is very important for plant cell, as we established before. So let's start from the top this time. What's the importance of carbohydrates in the bodies of living organisms? First, glucose is the way that the carbohydrates are transported all around our body. And we established before that glucose has to be transported all around our bodies for cellular respiration in order to get energy, right? So we need the glucose molecule in order to make immediate energy. So glucose molecules or carbohydrates, these are the immediate sources of energy in our bodies. Not only us, plants, any living organisms, they need glucose for cellular respiration and to get energy. So how can we store this very important source of energy? As animals, we store it in the form of glycogen. So glycogen is a carbohydrate, which is a polymer, a big molecule, in which we store a lot of small molecules or monomers. We put them together to make a glycogen. Where do we store it? We store it in the liver and in the muscle cells. Okay, why do we need to do this? To decrease the amount of glucose in the blood, because we cannot leave glucose running around the blood more than our use. What about plants? How do they store glucose? They store glucose in the form of starch. So starch again is another carbohydrate that is made from monomers or monosaccharides that are put together to get stored. Okay? So How do we test for the presence of carbohydrates? First, as we just learned, we have simple carbohydrates, that's glucose, or complex carbohydrates like starch, and cellulose, and glycogen. So we need two tests, a test for the simple sugars or reducing sugars, and a test for the complex sugars. For the complex sugars, we use iodine. Iodine solution, which color changes from brown to blue-black. Brown to blue-black. So when it's positive, it changes from the normal color of iodine, which is brown, into blue-black. In case of the presence of simple sugars, like glucose, for example, we use the Benedict solution. The Benedict solution is blue in color, and it's also heated till 80 degrees, and the number is very important. So when it's heated after being added to simple sugars, it's gonna change gradually from its original color, which is blue, to green, yellow, and then to orange red, and then finally to brick red. So the test is positive, which means that you have a reducing sugar when the color changes. What if no change happens? This means that you don't have any reducing sugar in this food. Benedict solution contains copper sulfate, which makes it blue or gives it its blue color. So when heated in the presence of reducing sugar, this copper sulfate breaks down into copper ions, which are reduced into insoluble brick red copper oxide. Okay, so the color changes from the blue color into the brick red color. And that's all about carbohydrates. Let's just remember or wrap it up. Complex sugars are tested by iodine or tested for by iodine. We know that there are complex sugars in the food. When we put iodine on them, what will happen to iodine? It's going to change its color into blue-black color. So you can remember it by CIB, complex sugar, iodine, blue-black color. In the Benedict solution, you use it to test for reducing sugar, and the result comes in brick red. So Benedict, reducing, BR, and the result is brick red, BR also. Okay, let's move to fats and oils, or lipids. So fats are usually solid in room temperature, but oils, they are liquid. So usually fats come in a solid shape, but except for oils, they are liquid. What are the elements that make them? The elements that make them are carbon, hydrogen, and oxygen. But actually, we have a very small amount of oxygen compared to carbohydrates. Okay, so basic units are 3 molecules of fatty acids and 1 molecule of glycerol. Again, 3 molecules of fatty acids and just 1 molecule of glycerol. They are insoluble in water. Insoluble means that they do not dissolve in water. So they produce a huge amount of energy. So what's the difference between them and carbohydrates? The difference is that their metabolism or their breaking down takes very long time. Carbohydrates, their metabolism is immediate. So they give us an immediate source of energy. But what about fats? They act as a storage for energy. So now we know one of their importance in the bodies or in the living organisms. What is another importance? The other importance is that they form the cell membranes. And actually, since they are insoluble in water, they create a layer. And this layer insulates the body and reduces the water and the heat loss from the body. And that's why they are called the waterproof layer. Also, they cover skin, feathers, or fur. That's why we have glands that produce oils to protect our skin all the time. And then, how do we test for them? We test for them by ethanol emulsion. So, lipids do not dissolve in water, but they do dissolve in ethanol. So, what do? We mix the food in ethanol, and this will allow any fat to be dissolved. And then we pour it into a tube of clean water. What will we see? This is what you will see, a milky emulsion. So remember, mix food with ethanol, M-E. You will get a milky emulsion. So fat me. Remember it by fat me. So when I mix food with ethanol, I will get a milky emulsion. This is to test for fat me. And let's move to proteins. So proteins, they contain 4 elements: carbon, hydrogen, oxygen, and nitrogen. And also some proteins contain sulfur. What are they made from? They are made from long chains of amino acids. So amino acids linked together will give us a protein. There are different kinds of amino acids. To be exact, 20 different kinds of amino acids. Their different combinations together will give you endless number of proteins. So each kind of protein has a different arrangement of its amino acids. And that's why you have a lot of proteins again. They are soluble in water. They dissolve in water. They like water. So what are the importance of proteins in the bodies of living organisms? First, the formation of enzymes and hormones. So proteins make all of our enzymes and all of our hormones. And of course, these are very important chemical messengers as hormones, or they make important functions in the body like enzymes. Most of your digestion process, for example, depend on enzymes. Formation of cell membranes in all organisms. But we said before that cell membranes are made of lipids. Yes, the main structure of the cell membrane is made of lipids, but there are very important protein channels and pumps, remember? These channels and pumps are made of proteins. Without them, the cell membrane will lose a huge part of its function. Antibodies that protect the body against diseases or pathogens. So antibodies, they form the army of our body to protect us from any invasion. What are they made from? Again, they are made of proteins. Proteins are also responsible for the growth and the repair of our tissues. Also, our hair and fingernails are made of proteins. Protein keratin, to be exact, is the protein that makes our hair and fingernails. How do we test for the presence of proteins? The test for proteins is the burette, which is simply potassium hydroxide, then drops of copper sulfate. They are carefully poured on our sample. And if the color changes from blue to purple or lilac, this means that it's a positive test. Positive means that it contains proteins. So, burette tests for proteins. How do we know it's positive? When the blue color changes to purple. So, BP. Burette proteins, blue to purple. And then finally, the test for vitamin C. Again, where can we find vitamin C? It's found in many fruits and vegetables, especially the citrus fruits like oranges and lemon, and in leafy green vegetables as well, such as cabbage or potatoes. What's the importance of vitamin C in our bodies? It helps to keep our skin and hair healthy. And we will learn more about the importance of vitamins and minerals in later topics. So how do we test for the presence of vitamin C? We use a test called DCPIP. We add the DCPIP, which is blue in color, to the food sample, and if it's positive, the blue color will disappear in the presence of vitamin C. But what if it's a negative test? The color remains the same. So DCPIP is a test for vitamin C. If it stays blue, this means that this sample does not contain vitamin C. But if the color changes or disappears, this means that this sample contains vitamin C. So let's look at some questions. What is the color change shown by Benedict solution when heated with a reducing sugar? So when you heat the Benedict with a reducing sugar, what do you get? Benedict reducing BR. So the color will change again, BR, from blue to red. So the answer is B. The table shows the result of food tests carried out on a fruit. So scientists had a fruit and they wanted to know what are the food molecules that they can find in this fruit. So they tried the different tests on it. Benedict, it came out positive. So let's remember, Benedict was the test for what? BR. Benedict was the test for reducing sugar. So please in the exam write up here reducing sugar. And then burette, BP. Burette, protein, from blue to purple. So please write here protein. Ethanol was the test for fat and iodine was the test for complex sugar. So these came out negative. So I'm looking for the choice that contains reducing sugar and protein. So now the answer is very easy, right? It is C. Okay, the next question. 4 solutions were tested to see whether they contain protein, starch, or glucose. The colors of the solutions after tested are recorded in this table. Which solution contains protein and glucose but not starch? Okay, in order to answer this question easily, I will go to protein and write under it positive burette. Starch, I will write positive iodine. And glucose, I will write positive Benedict. So I will write then the color. Positive for protein, purple. Positive for starch, blue-black. And positive for glucose, it's gonna be brick red. Now I will continue reading the question. What does it say? Which solution contained protein and glucose, but not starch? So not starch means that which solution or which answer or which test should be negative? Yes, the iodine test. So the iodine test should stay brown as is, while the burette should change to purple and the Benedict should change to brick red or orange. Of course, orange will be accepted because it was the intermediate stage. So let's try solving it. First, I need to make sure that the protein and the glucose go positive. So I need to cancel the iodine or the starch. So of course, this test or this choice will not be chosen because blue-black is the positive for iodine, as I wrote up here. And also, D cannot be the answer because I wrote up here for myself that if starch is positive, it's gonna give me blue-black. And the question says that the starch is negative. So I already knew that B and D are not the right answers. So it is C. Why? Again, because I have negative iodine test, yet I have the purple for the burette and the orange or the brick red or the yellow for the Benedict test. So this is how we answer such a question. Let's take a look at paper 3 questions. Choose the words or phrases from the list to complete the sentence. Each word or phrase may be used once or more than once or not at all. This is very important. Most of you ignore this part of the question, and that's why they feel like there's something wrong in the question, or there are a lot of words that were used more than once. Please read everything in the head of the question. Fats are composed of these elements. What are fats composed of? Fats are composed of carbon, nitrogen, and oxygen only. All proteins contain 3 elements, and the element nitrogen in addition to those elements. Glucose is a type of carbohydrates. Animals store excess glucose as glycogen in the liver, also in the muscle cells. Plants store excess glucose as Starch. Also, plants use glucose as cellulose to make their cellulose. Thank you, and I hope this video helps you study.